Welcome everyone to our virtual 2021 PWS Family Conference presented by Soleno Therapeutics. Before we begin today's presentation, let's first thank our amazing sponsors, Soleno Therapeutics, Levo Therapeutics, Harmony Biosciences, Radius Health, Inversago Pharma, Saniona, Consonance Therapeutics, and Novo Nordisk. Their support allowed us to make this conference free for all of our community members, and we are incredibly grateful for their contributions and their support. In today's presentation, Dr. Theresa Strong, FPWR's Director of Research Program, will be providing highlights from last week's Research Symposium, as well as highlighting a few of the grants that are funded through FPWR's research program. Many of you are already familiar with Dr. Strong, but for those of you who don't, she's not only our Director of Research Programs for the last decade, leading our research team, but she's also an accomplished geneticist and a mother to a young adult with PWS. It would be an understatement to say that Theresa is my hero. Her personal contributions to this organization have been numerous, and we would not be where we are today without her. So Theresa, I hope I haven't embarrassed you, but you are an amazing asset to our community, and the floor is now yours.
Well, thank you, Susan. I may embarrass myself by not being able to share my screen. So let's see if we can do that after that, wonderful introduction. I really appreciate it. So welcome everybody. As Susan said, my task here today is to give you some information about the research symposium that we held last week. And that was also a virtual research symposium for the second time now, the second year. And we really look forward to getting together next year in Chicago in person. Yeah, I think it was a great conference, but just like these virtual conferences, it's always better to be together in person. So this conference took place last week. There were 2 days of talks, 21 talks in all. There were general sessions, and then we had concurrent sessions with a clinical track and a molecular biology track. We also had a virtual poster session, so scientists present their work as posters a lot at meetings, and we had about 16 posters, and so those were available for viewing, but we also asked all of our presenters to do a short recording where we kind of walked people through what was on the poster, and that was available to the scientists as well. So that hopefully was a good way to get those other projects communicated. Because we were virtual, the one, I think, advantage of that is that it was an international meeting. So we had more than 9 countries represented and presenting, which was fantastic. Just a shout out to our friends in Japan and Australia who were effectively getting up at 2 or 3 o'clock in the morning to join us and be present for the question and answer sessions. So there's some real dedication there in our scientists. And it was a great mix. One of the wonderful things about these conferences with the focus on PWS is you really get a broad group of individuals. So there are PhD scientists, bench scientists, looking at molecular things. There are clinicians who are looking at standards of care and how to improve care of a person with PWS. And so there's so much learning between the groups. I think that is one of the really valuable things of a conference that is focused on a rare disorder like PWS. So we saw senior investigators, we saw trainees presenting their work. It's always great to see the young scientists being exposed to PWS because we hope that they'll stay engaged in the PWS field for their careers. And the FPWR research team also had a couple of poster presentations. We spoke about this a little bit in the session on Tuesday morning about what the team is doing, but we felt it was important to share this with the scientific world that we're looking at the PWS genomes, whole genome sequencing of individuals with PWS and linking that up with the data in the global PWS registry. And Lisa Mattisbach presented some work on some of the early findings of the Path for PWS study, specifically with respect to medication usage. The abstract book from the scientific conference is available now on the FPWR website. You can go to the link and you can download that if you just like to sort of peruse. But my task here today is to try to hit some highlights of that I really enjoy talking about science. There were a lot of presentations, as you could see. I'd love to talk about it for hours, but Susan won't let me. So what I've tried to do is really categorize them and then pick one or two presentations in each category to speak about. So the areas that we're going to cover are molecular genetics and cell biology of PWS, the insights that we're getting from the animal models, some of the clinical studies that are either done or planned, and then genetic therapy to wrap it up. So, you know, one question sometimes people have if there's, you know, not from a scientific background is, you know, why are we as a foundation supporting these very basic science studies to understand PWS genes? You know, why don't we just develop therapies? Well, I think, you know, the answer to that is that you can be much more efficient and effective at developing therapies if you understand what is different in PWS compared to typical cells. So we really want to understand, what do the genes in the PWS region normally do? And then how, when those genes are lost, do we get the symptoms that are associated with PWS? If we can understand that at the molecular and cellular level, then we can identify targets for therapy. We are developing therapies. There are drugs in clinical trials, and they're based on some understanding of PWS, but I think we have a long way to go before we could confidently say that we really understand all the pathways that, for example, lead to hyperphagia, lead to intellectual challenges, and really be specific about targeting those specific pathways. So I feel like, you know, the importance of this work is to allow us to develop therapies more effectively and more efficiently. So I'm gonna show you the PWS region again. So I know not everybody wants to be a geneticist, but I feel the need to make everybody a geneticist. So I'm gonna just, because, well, I'll be using the names of the genes, and this way maybe it will make a little bit more sense to you. This is chromosome 15 shown across the top. And if we blow up this region, this PWS region in chromosome 15, all of the genes shown in blue, all of the blue dots, are genes that are expressed only from dad's chromosome. And so these are the genes that are lost, we lose expression of in PWS. And there are some sort of more typical genes. These ones that have the little stars are, they make proteins. And then there's some more unusual genes, these ones that are these ovals are, they make RNAs, and those RNAs never make a protein, but presumably they regulate other RNAs. So we know a bit about this region. There's been a lot of work over the past 25, 30 years since this region was really defined. We know some of these genes, for example, the McCoran-3 gene all the way on the left here is important in puberty, so triggering that puberty. So it makes sense. We see incomplete sexual development in PWS, and so that may underlie that. We know the nectin gene is related to the gene right next to it, MAGL2, and that it has a role in helping neurons differentiate. And it also, when we lose just that gene, there's some respiratory differences. So this may underlie some of the sleep disorders that we see in PWS. But one of the most important genes in this region is the MAGL2 gene, and you'll hear a lot about that. And it's important for many reasons. One reason we know it's important is because if you have a mutation just in MAGL2, so if all of the other genes in the region are fine and are normally expressed, what you get is Shafiang Syndrome, which has a lot of commonality with PWS. And we'll talk about that. That was one of the presentations. So we know that losing MAGL2 probably is a big contributor to the PWS phenotype or characteristics. We also know this RNA over here, SNORD116, is critical in PWS because every person who has the symptoms of PWS has lost SNORD116. And that's where it gets really complicated. There are some really funky, there's a RNA transcripts, and that they're processed into smaller transcripts. And experimentally, it's very difficult to tease out, which of these processed fragments is doing what in the cell. And that's where a lot of the scientists have spent a lot of time is sorting through that. So for the abstracts in molecular genetics and cell biology, there were quite a few. Dr. Weverick, who has worked in PWS for many years, she's on our scientific advisory board. She's done some great work with MAGEL2 and Nectin. She was looking at the MAGEL2 protein and trying to characterize that better. What their group found is that there's the first part of the protein has a structure that's called intrinsically disordered, which is a great word. I kind of felt like it describes my life some days, intrinsically disordered. But that structure, figuring out what that structure is really important for helping us understand what the protein may be doing. And she and her lab, they've also looked at what proteins are associating with that part of MAGEL2, and they found that MAGEL2 may have a role in RNA metabolism. So previously, other groups have shown that MAGEL2 is important for moving vesicles around cells. This may just be an additional role that MAGEL2 has. There were two studies that were, again, really complex molecular genetics, so I'm not going to go into them very much, except to say there are some very fantastic groups that are working on this problem of what those different RNAs, the long one and then the SNORD 115 and 116, what exactly they're doing within the cells. And this was a group from Scotland, as well as the University of Birmingham in England. I want to talk in a little bit of depth about some work from Larry Ryder's lab that was presented by Caitlin Victor, and she's one of sort of the young investigators that we're really excited to see in the lab. Many of you are familiar with this project. We've supported the Ryder lab for a few years now because they've been doing really interesting and unique work. So they use baby teeth that have fallen out of the mouths of our kids, and they're able to extract stem cells from those baby teeth and then grow them into neurons in a dish. So it's a great source of neurons from someone with PWS without having to take neurons, for example, from their brain, right? So it's just a great resource. Many of you have sent Caitlin and Dr. Ryder your kids' baby teeth. I think they're always looking for more, so you can contact the Ryder lab and get that set up. If you have a child who is likely to lose baby teeth soon, you have to get the media ahead of time. But they've been looking, so they've been differentiating the neurons in PWS, and they've gotten such a nice panel of baby teeth now from kids with deletion, from kids with uniparental disomy, and then kids with and without autism, as well as PWS. And so they're asking, what are the molecular differences between those groups? And can we identify new targets, particularly for our kids who have PWS and autism, that might be able to help that? So they presented about the gene expression changes that they see when they compare any PWS neuron to any typical neuron, and then again, the subgroups. They identified some cellular and molecular changes in deletion versus UPD, and then in UPD plus minus autism. And one of the interesting things that they found is that the mitochondria seem to be different in kids with autism versus kids who do with PWS but without autism. So this just shows you on the left a picture of a different cell, and the mitochondria are stained. You may remember this from high school biology, right? Mitochondria are the powerhouse of the cell. And what they found that there were differences in the number, the volume, and where the mitochondria were localized within the neurons of those kids who had autism versus those kids who did not. So this is a really interesting line of investigation, and they're following that up with some additional FPWR funding. They're going to be looking at the bioenergetics of mitochondrial function in PWS neurons. They're going to look at some specific genes that they think are downregulated in those who have ASD. And these are genes that are important in making mitochondria and keeping them healthy. And interestingly, there are already some drugs that target these pathways. So they're going to try out those drugs in the neurons from the baby teeth. And the other thing that they're going to look at, which I think is a really interesting line of investigation, is the specific changes in our kids with UPD compared to our kids with deletion. So we know our kids with UPD who have 2 copies of mom's chromosome 15 may have higher levels of expression of a particular gene that is only expressed on the mom's chromosome UBE3A. And having higher levels may not be a good thing. And so what they're going to do is use molecular techniques to reduce the levels of UBE3A and then see how that impacts the cells. And that may, again, give us some ideas about new ways to target some of the specific challenges that we see in different subtypes of PWS. Moving on to some of the insights from animal models of PWS. As you know, we have a lot of rodent models of PWS, mice, and now a rat model of PWS. And there's been a lot of work using those models to better understand PWS. We have some challenges in that way. The mice they don't fully recapitulate the characteristics. They don't typically get very overweight. They don't have the same food drive that we see in humans. But nonetheless, there's a lot of features that they do have and a lot of ways that those models can be used to better understand PWS. So one study came from Anthony Isles, who is a fantastic animal model researcher in the UK. He has worked in the area of PWS for a long time. He is a member of our preclinical animal network. And he has a lot of expertise in PWS mouse models and PWS behaviors. And he's specifically looking at the mouse models and their behaviors and seeing if he can gain some insights into the more serious mental health issues that some of our kids will have as they reach adulthood. And he's specifically marrying the behavioral modeling in the mouse with some genetic data from humans that have mental illness, that have different kinds of mental illness, and seeing where they merge to try to, again, define those pathways that are present in PWS that make someone with PWS more susceptible to serious mental health issues. So it's a really fascinating, and I think, he's one of the few people in the world who could kind of bring that all together and use those insights for this problem. We also heard from a group at Yale University, the Dietrich Lab. Gabriella presented about the MAGEL2 mice and showed that, so this is a PWS mouse model, and mice, well, mice squeak, but they actually have ultrasound that they do to call their mothers to them when they're little babies. And the PWS mice don't do that. And that causes a sort of a disruption of that maternal caring for the baby. So that was a really new and interesting insight into sort of the social communication. And it paves the way for maybe being able to look at interventions for that in the human situation. We also heard from Liz Malecki-Bass, who's at University of Buffalo, and she is looking at the new PWS rat model that FPWR funded. And by the way, on these slides, not everything that I'm presenting was funded by FPWR, but I've tried to indicate the studies that were funded by FPWR. We have funded Dr. Isles for different work, but the work that he presented here was funded by, I believe, federal agencies. Anyway, looking at the rat model of PWS, that is a newly generated model. And so a scientist is an expert in looking at energy balance and feeding behaviors in rats. And what she's found is, which she'll have to follow up on because it's early days, is that the PWS rats seem to, they don't look too differently. They don't get grossly obese. So similar to the mouse model, they don't, overeat to the point of becoming obese, but they do hoard their food. So they have some unusual behaviors around food that might be interesting. And again, might offer some ways to, to look at drugs that might help with some of that obsessiveness about food. We also heard from a pair of researchers who have collaborated quite a bit over the years, Dr. Muscatelli, who's an expert in PWS mouse models, and Dr. Bourret, who is an expert in neurobiology. And so they have worked together to, with Dr. Muscatelli, generating mouse models, characterizing them, and Dr. Bourret really looking at the brains and the neuronal development in those animals in many different aspects. But in particular, what they presented on was how to understand the changes in oxytocin and PWS and how to manipulate that sort of to its best effect. So this is kind of laying the groundwork for using oxytocin or carbetocin, for example, in the best way possible for individuals with PWS. So these investigators have shown that there's a deficiency of oxytocin neurons in both mice and humans with PWS. And in mice, if you give that oxytocin back shortly after birth, that improves social cognition and behaviors. And that work, along with other collaborators, has led to some studies by the company OT4B administering oxytocin to newborns. And in this case, they're examining how oxytocin is needed and how it functions across the lifespan. So to understand, are there only benefits in newborns? Are there benefits later on in life? And are those the same and do they differ? So they've been using a genetic toolbox essentially to turn oxytocin neurons on and off in these mice, and then look at how that impacts development and metabolism. They looked at inactivation of oxytocin neurons at birth or in adolescence leads to autism-like behaviors. So that suggests at least that restoring oxytocin at those points might lead to better social outcomes. They've also shown that oxytocin is important in a particular system that regulates metabolism in the hypothalamus. And they've also shown that if you take juvenile mice and inhibit oxytocin at that point, that it increases the fat mass. So there's both effects on social interaction and cognition, as well as effects on metabolism and body fat, and those things seem to happen at different times. So this work is it's definitely ongoing, and it is really aimed again at teasing out when oxytocin is going to be most useful for what aspects of PWS. Okay, so I'm going to move now to the clinical sessions. There were a lot of abstracts in the clinical sessions. We heard about completed clinical trials, one of which you heard about yesterday. So Levo Therapeutics presented at the family conference yesterday. You'll hear from Soleno Therapeutics today about their drug DCCR. So both carbetocin and DCCR have completed phase 3 clinical trials. And that was presented to the scientific community as well as to our families. We also heard about planned clinical trials from Radius Health using a synthetic CBD oil oral solution. And you may have heard about that in the clinical trial session yesterday. Consonance presented about their novel triple reuptake inhibitor. So this is a drug that impacts the serotonin, norepinephrine, and dopamine systems. I think I have that right. Oh, there it is right there. I guess I could read it. Affecting those systems, and they have a particular, a very individualized dosing scheme planned for a planned clinical trial in PWS. And Dr. Komonis talked about administration plans for a clinical trial of oleoethanolamine, which is an endocannabinoid-like lipid molecule, and she is working with a company to bring that to a clinical trial in PWS. So some clinical trials completed, some clinical trials planned. Perhaps a little bit less glamorous, but very important, is developing tools to do these clinical studies. There were a couple of presentations about the degree of hyperphagia for example, in young kids with PWS age 3 to 8 compared to other neurodevelopmental disorders, where they found the kids with PWS, not surprisingly, had more hyperphagic behaviors. But some kids with Angelman syndrome, which is also located in the PWS region, also seem to have some hyperphagic behaviors. And there was a study from Dr. Gallagher's group in Ireland looking at a sort of a computer-based task looking at food to see if that might be a way to measure attention to food and might serve as a clinical trial outcome measure. We also heard a good bit from Dr. Christian Schaff, who is Schaff of Shafiang Syndrome. So he is the physician scientist who first described Shafiang Syndrome, and he has been working on it since its description almost a decade ago. And he's been working both on the clinical side and on the molecular biology side. And one of the key things that he's doing is comparing how do individuals with Shafiang differ or are the same to individuals with PWS. And Shafiang was, initially identified in just a handful of individuals. That pool of patients has grown over the years as more people are doing whole genome sequencing, but it's still a small number of individuals. So there's still a lot of learning to be done. I'll mention that Dr. Schaff did give a talk to the Shafiang families. He's probably much better at talking about all of this than I am. So I think that recording will be available and you may want to look at his talk if this is of interest to you. But what he found, what has been described is that when the MAGEL2 gene is kind of cut off, when it has a mutation that stops it, you get Shafiang syndrome. And he's been looking at the overlaps between the phenotypes, again, between Shafiang and PWS, and he has found a lot of similarities. So when you mutate just MAGEL2, you get hypotonia at birth, feeding difficulty, sleep disruption, growth hormone deficiency, other hormonal changes that are very similar to PWS. There are some unique things about Shafiang compared to PWS. Individuals with Shafiang oftentimes have contractures where their muscles tighten up and can't easily open. They may have more severe intellectual disability and a higher rate of autism. Why that is an area of active investigation. For example, the work that Rachel Weverick is doing, the work that Ryan Potts has done is beginning to shed light on that, but it's still a bit of work in progress. Initially, when Shafiang was described, Dr. Schaff made the distinction that there doesn't seem to be very much obesity and hyperphagia in Shafiang syndrome, and that was thought to be a difference. In fact, as more patients have come to light and Dr. Schaff has seen more older patients, he's changing his thinking on that in fact, as kids get older, they do seem to have more food seeking. They do seem to be very easy to gain weight like our kids with PWS. So there seems to be more and more overlap between these two symptoms, again, indicating that MAGEL2 is particularly important in our PWS region. Dr. Schaff has also, again, he also does molecular biology, and he has looked at a particular pathway, the MTOR pathway, and seen that it is upregulated, so it's high in Shafiang syndrome and PWS. So MTOR is a protein that's a component of a key cellular pathway that's important for nutrient sensing, so telling what kind of nutrients are around and then regulating cell growth accordingly. So it's cell growth and homeostasis, just kind of keeping things in the zone where they need to be kept. So he's identified that mTOR is upregulated in Shafiang and PWS, and that's not really a situation where we want. mTOR pathway is kind of a Goldilocks thing, right? You don't want too much, you don't want too little, you don't want it overactivated, you don't want it underactivated, you want it kind of in the range of just right. So there are already drugs available that will modulate mTOR levels, and he is using one of those drugs, rapamycin, to look at, he's already looked in some cell models and shown that it brings mTOR levels back to kind of a normal level. Now with FPWR funding, he is testing that in mouse models, and particularly this pathway, it's it's important throughout the body, but it's particularly important in muscles. And so as a readout, he's looking at the strength. We know that the MAGL2 deficient mice, they're kind of couch potato mice. They don't like to run. They like to sit around. They don't like endurance exercise. They're not marathoners. And so he's seeing if bringing the mTOR levels down will help those aspects of metabolism and endurance in mouse models of PWS. This is exciting work because it does have the potential to translate fairly rapidly into the clinical situation. There is a lot of drug development. There are some drugs already, and then there's new drug development that impacts the mTOR pathway. So one more clinical study that I think is really, it's a little bit different from all the molecular biology, so I'm going to move away from most molecular biology now, was presented by Elizabeth Dykins, who is a long-term researcher in the PWS field. She has really focused her career on helping families with neurodevelopmental disorders, including PWS and other Williams syndrome and things, have a more fulfilling life, like really focusing on those quality of life things. And so she has done a lot of work understanding the behavioral differences in PWS and the social cognition differences, so why it's difficult for our kids to make friends and keep friends. And she's really interested in improving that, making that better for our kids and for all of us. So social isolation and loneliness are very common in PWS and in all individuals in the intellectual disability category. It just, I mean, it's common across society. It tends to be more common in some groups. And people with PWS may have challenges in making and keeping friends because, you know, they're not necessarily, they have some challenges reading social cues. They tend to focus on their things and they may be perseverative. They may have temper outbursts, they're rigid thinking, and that makes it harder to be a friend. So Dr. Dykins and her team are really interested in, you know, can we work with young adults with PWS and help them get better skills so that they can improve these aspects of their life? And this was a Zoom-based intervention, and they were cool with Zoom. It was before the pandemic, so they even know how to do Zoom prior to the pandemic. So they call the program Building Our Social Skills. And, you know, it really looked at recognizing emotions in others, regulating your own emotions, and then some skills, some really practical, concrete skills, which are great for our kids for developing and maintaining friendships. And in this presentation, Dr. Dykins, you know, presented some of the outcomes. She saw robust, or their team saw robust improvements in social cognition, awareness, social awareness, and communication. Both the participants and the parents described more friends after the Boston intervention. Participants expressed less feelings of loneliness, and clinicians who were rating the social skills of the participants saw some improvements. So, you know, a disclaimer here, my son participated in this program. I thought it was great for him. You know, he wasn't, it took him like 1 or 2 sessions to get engaged, and then he really enjoyed it. And I could see him practicing some of the practical skills that he was learning there, which was great to see. So the next step, the team at Vanderbilt is interested in developing a curriculum so that this can be, you know, done more widespread and benefit more kids with PWS and perhaps other developmental disorders as well. I think finally in the clinical session, I'll mention a newborn screen that has been developed by David Godler with funding from FPWR and also from some other neurodevelopmental disorders that are included in the test. So Dr. Godler has developed a test that can detect Prader-Willi syndrome, Angelman syndrome, and duplication 15 syndrome in one test in a blood spot. And so, you know, why would we want to do newborn screening? So this is the screening that happens to newborns, you know, in the nursery, the sort of the heel prick and the blood spots. We know that babies with PWS are not always identified at birth. Probably 20–25% of kids with PWS still are not getting picked up in that newborn period. It's really dependent on the physician who's in charge. If they're familiar with PWS, if they suspect PWS, then they'll order the right test. But if they don't, they may not. And so there's probably certain groups, rural areas where, a physician maybe hasn't seen someone with PWS. Ethnic minorities that don't have the typical, we hear in PWS because of one of the genes that's lost, many kids with PWS are blonde hair, blue eyed. Well, obviously for some, other ethnicities, that's not going to be the case. So there may be certain groups that are really getting underdiagnosed. We have very good evidence that early intervention and growth hormone therapy improves the outcomes for individuals with PWS, and you have to have a rationale about why you would do newborn screening in order for states to consider doing newborn screening in the US. We don't know the true incidence of PWS. So pharmaceutical companies come to us and say, okay, so what's the birth rate of PWS? Well, we can make a guess, but we don't actually know. Newborn screening would allow us to actually know, have all those individuals identified at birth, and then put them right into the early intervention that they need. So a molecular test to detect PWS with a greater than 99% accuracy is available. The challenge is being able to do it on a scale of thousands and do it for a cost that is feasible. You can't have a test that is $200 per baby, just economically, it's not going to work. You have to get the costs down to a very low level for it to be considered to be implemented in newborn screening. So Dr. Godler presented about, the assay that they've been using and his procedures to, do the screening in a high throughput, confirm the ones that look like they're positive. He was able to bring the costs down to a reasonable level, $1 per assay per disorder. And he screened about 16,000 blood spots in Australia, this is, and identified 2 babies with PWS, 2 with Angelman syndrome, and 1 baby with Dup 15. It suggests, although it's only 16,000 samples, that the incidence of PWS and Angelman may be a little bit higher than that 1 in 15 to 1 in 20,000 that we hear. But we don't know until we expand it further. And that is, the next steps is to expand this pilot in other countries, including the US, and see if it's feasible to implement in. There is a very complicated procedure for adding something to the newborn screen in the US. There's a lot of steps, and it's going to take a lot of collaboration with many of the physicians, not only in the PWS field, but also in the Angelman and Dup 15 fields. But we are working very actively with those groups, and keep your eyes open. We hopefully will be moving forward soon with that. Okay, last little bit, I'm going to talk about gene activation because that's always interesting, I think, for all of us. Again, in the normal situation, the PWS genes are only expressed from the chromosome that came from dad. But all of our kids with PWS, whether it's PWS by deletion, PWS by UPD, or by imprinting, they all have the genes sitting there on the mom's chromosome. They're just sitting there, but they're silent. And so a lot of the work is trying to activate those genes, asking whether they can be awakened, and then asking the questions, would it matter if they are? And at what point during life would it have to be done in order to have a positive effect? So feasibility, technical concerns, and then, practicality and impact are all questions that are outstanding. And we have many investigators who are working in this area. Here are just a list of some of the recent FPWR funded projects that are looking at these questions. And a couple of these were at a point where they could present some data at the meeting. So a group in Germany is looking at it actually was a very technical, very good, but very technical talk. There's a lot of different tools. You've probably heard of CRISPR. There's also these zinc finger proteins and what's called talons, different molecular tools for turning genes on. And this group is kind of running through combinations of those and trying to optimize it specifically for the PWS region. There was also a gene therapy talk that was a little bit of a different track. So not looking at gene activation, but really looking at one of the downstream pathways that may be affected in PWS, the BDNF gene. So it looks like BDNF is decreased in PWS as a result of whatever molecular changes are upstream. And so this group is asking, if we replace BDNF and get it sort of fine-tuned to the right level, will that improve metabolic function and some other behavioral aspects? And they're doing that in a mouse model of PWS, and they're really making some great progress. They've already shown that they have some impacts on the MAGEL2 model. We've recently funded them to continue the work to see if that is a potential therapeutic strategy. And it's really about defining whether BDNF pathway is a good pathway to target in PWS with gene therapy or with other approaches. The final talk I'll mention is the work of Marnie Blewett, who is who is in Australia, who we have funded, who's also been funded by the Prader-Willi Research Foundation of Australia. We've kind of worked together on that. She's doing some really interesting work on this protein called Schmidge D1. And this protein is known to be one of the important proteins in keeping the mom's chromosome 15 silent. And it's specifically in this cluster, right? So the genes in the mouse include PEG12, McCorin, MAGEL2, and nectin. And Schmidge D1 silences that on the maternal allele. And so the concept is, what if you got rid of Schmidge D1? Could we reactivate those genes? And would there be other, because Schmidge D1 also inactivates other genes, and so what would be the off-target effects of that? So as proof of concept, she's shown that when you delete the Schmidge D1 gene, you do get expression of the PWS gene cluster. Interestingly, again, it's these genes. It does not include the SNORD116 gene. And so in a way, it may be a really interesting system to understand what is the impact of MAGEL2 and nectin only in the absence of SNORD116. We don't know if you took a person with PWS and you gave them MAGEL2 back and nectin and McCorin, how far would that go to correct some of the deficits that we see in PWS? So with previous FPWR funding, Dr. Blewett showed that if you eliminate Schmidge D1, the PWS genes will turn on. And then she's extending that work, looking at different approaches to see if inhibiting Schmidge D1 would be a safe and effective approach to activate expression of MAGEL2 and the other genes. When would that have to be done again, and what would be the impacts? Can it be turned off without having a negative effect? So far, that looks good. When would it need to be done? They're starting to investigate that. Because most of their work was in mouse models, they're also now looking at human cells to make sure that the same things are occurring in human cells compared to mice. And so far, it seems to be holding up well between mouse and human. Dr. Blewett's group with other funding is doing a high throughput screen. Schmidge D1 is an enzyme, and enzymes are considered druggable targets. So she's doing a high throughput screen where they screen thousands of compounds and try to get one that is very specific for Schmidge D1, so that maybe you could use an oral drug to turn Schmidge D1 off at certain times and allow MAGEL2 and the other genes to come up. So really interesting work there and great progress. So, so you've stuck with me for a long time, which I really appreciate. If you want to stay up to date, we have new projects that are coming. We're going to be giving a webinar about that. I've given some hints through here, but we'll talk about it later. We have several. We have a page that talks about outcomes from our FPWR funded projects, and they're listed here. You can always contact me, Jessica Bahanowicz, or anyone on our research team with specific questions, or if you have specific things that you'd like to hear about. Please contribute to the research. If you have a loved one with PWS, consider joining the Global PWS Registry. That is such an important resource. We learn so much from everyone who is involved there. Please consider signing up for the PWS clinical trials alert. Even if you are not at a point where you want to enroll in a clinical trial, we also will send out requests for baby teeth or, other, one time we had a request for poop from kids with PWS, and everyone has poop. So, there are, even beyond clinical trials, which we all hope you consider enrolling in clinical trials, there are other opportunities to be advancing PWS research. So, and lastly, I really have to thank all of our donors. So much of this research would not have been possible were it not for the support of our community and our donors. It has been fantastic to see how this field has grown. And I think FPWR has had a critical role in identifying those projects, identifying fantastic researchers to come into the PWS field. And we can only do it because of the financial support and time and energy that all of you put into sending baby teeth and doing your registry and participating. So thank you so much.
All right, so at this time, we can go ahead and move into Q&A. We do have a couple of minutes available for some questions. Again, you're welcome to submit your questions through the Q&A panel, and we will get through as many of the questions as we can. Theresa, in regards to the BOSS program, you mentioned that a curriculum is being developed. Will that curriculum be able to be used by parents, or will it require a professional?
Well, that was one of the things, and I think it would be great if we could, share Elizabeth's presentation, because she talked about that. One of the things that they looked at was, they had, so if you know the group at Vanderbilt, Elizabeth Roof is, really experienced with PWS, and Haley is Hunt Hawkins as well. They did some of the sessions, but they also trained graduate students to run other sessions. And what they found was the graduate students did fine. So that suggests that, teachers, people who are not necessarily experts in PWS may be able to apply this curriculum with great benefit. So I think that was very encouraging. I'm really glad that they did that. They had groups running in parallel and showing that it might be easily widely dispersed.
Mm-hmm. That's great. I know, social challenges are very prevalent, especially in our own house. So it'd be great to be able to have resources or be able to give a professional some resources as well. Lots of questions about, can individuals access Dr. Dykens' presentation? The answer is yes. All of the presentations are available. We'll be following up with an email after, give me a day. So everyone who registered for this session will get an email. And we'll link to the symposium presentation. So if you were interested in specific presentations that Theresa mentioned today, you can go and actually watch the full length presentation. They're only 15 minutes, so they're pretty short and easy to get through. Theresa, the Radius clinical trial, you mentioned that they're going to be testing CBD. How is this CBD different from what we can get online or at the grocery store or at a local market?
I would say that there is likely to be a significant difference. So the CBD that you get online or at your market is not regulated by the FDA. It's considered like a supplement, and that means that it is not monitored. So you have to kind of believe that what's on the label is what's in the bottle. In contrast, this is a CBD that has been made in the lab, so there's no contaminating. One thing we worry about is our kids with PWS are susceptible to psychiatric disease. And if you have THC, which is a component of some of these CBDs, other studies in typical populations have shown that makes young kids more susceptible to psychiatric illness. So that seems like something we don't want in PWS. The advantage of the Radius Health drug is that it is regulated by the FDA. So what's in it is in it, and somebody's kind of checking on that. And it is made in the lab. And so there's no chance that, some of the components that you might not want in a CBD solution will be in there. So I think it is just, the potency, purity, you have this list of things that you have to show to the FDA that it's the potency, purity, all of these things are the same. And so I think that you know what you have in the clinical trial. And the other thing is, if you want to think in sort of a community level, if everybody's using different CBDs and then, you're going online and saying this one works, this one doesn't, it's hard to know what actually works. In the context of a clinical trial, everything is given the same way, the assessments are the same, and we can probably get a better answer about whether this is, helpful in our kids or not helpful. So it's a little bit of short-term pain having to go through the clinical trial, but then really understanding the benefits and potentially the risks, right? I mean, so if you're in a clinical trial, they're going to be very carefully looking at safety. So that was a long answer, sorry.
Well, but you make up a really good point. If you're in the clinical trial, they're looking at safety, they're visiting with your child frequently. You get a little extra help versus trying to just test, an unknown substance on your kid at home. There was a question about what is CBD.
Cannabidiol, it's a derivative from marijuana plants, or it can be synthetically made as in the lab.
Mm-hmm. We had a comment from Elizabeth Roof. Hi, Elizabeth. She's joining us as an attendee today. She was part of the BOSS study, and she just wanted to mention, she hopes that people can be trained to deliver the curriculum. Parents may be harder to do it with their child since it needs to be groups of 5 to 8 people. One person with PWS on their own just wouldn't get enough exposure. So the program really requires a small group of individuals to practice the skills together alongside with a moderator. Let's talk a little bit about gene therapy. We have a couple of questions coming in there. Theresa, when you were discussing gene therapy, you threw out some terms, upstream and downstream. Is there a way of describing what that means?
I think I was talking about, so, we lose PWS genes, right? And then something happens that may cause other genes to go up or go down. So when I say upstream, I'm talking about sort of the primary issue, which is loss of MAGEL2, SNORD116, whatever, has downstream effects. That may cause, for example, the MTOR pathway to increase. So we can either try to fix the upstream problem, so get the genes activated or get them back, or we can modulate what gets changed down here. So that's 2 different therapeutic avenues that one can pursue. Mm-hmm. That was helpful.
No, it is. I have a visual in my mind, which I'm not going to describe for you all because it could be crazy. So there's always this question, which is gene therapy. Prader-Willi is caused by UPD, by deletion, by imprinting defect. How could gene therapy help all 3 of those subtypes?
So I'll just give you this picture again. The maternal chromosome, every kid, so Daniel has deletion, but he has my chromosome 15. My chromosome 15 that I gave him has a MAGEL2 gene sitting on it. It has a SNORD116 gene sitting on it, but they're silent. And so they don't make the RNA and make the protein and do the thing in the cell that they would normally do. Same for someone with UPD. They've got 2 maternal chromosomes sitting there. Neither one is active. The MAGEL2 gene is sitting there. It does not make an RNA. It does not make a protein. And so it cannot do its function. So regardless of which type you have, the genes are sitting there, but they're silent. We do have questions. If you turn on the gene in someone with deletion, you're only going to get sort of a normal level of expression. If you turn it on in someone with UPD, you may get too much expression. And those are all, that's this genotype specific issues. There may be differences when we turn these genes on in how we have to regulate it in someone with deletion versus how we have to regulate it with someone in UPD. But those are really questions kind of for down the line. First, we have to actually activate it. then we have to show that it has a meaningful impact. I mean, you could say, maybe you have to activate it during, fetal development. I don't think so, but we haven't shown that yet, that if we activate it in a baby or, an adolescent mouse with PWS, that it's going to have an impact. So all of those questions need to be addressed, but the genes are there in everybody.
Right.
And the other thing we can do is we can take the MAGEL2 and Nectin and we can try gene replacement, try just sticking them in there. Again, previewing some of the work that we're going to be funding, there'll be some strategies to do that as well.
Right. You mentioned we don't know at what age gene therapy has to be implemented. And the question looms, does it have to be done by a certain age? There are other syndromes where genetic therapy has been implemented at a later age and they have seen benefits, correct?
Absolutely. So, I think the thinking in the field 10 years ago was, look, it's a genetic disorder. The baby's born. There's nothing you can do, right? The brain doesn't change. It's a done deal at birth. And that thinking has really changed over the last decade. So we know that the brain continues to develop and that that you can, that it's plastic, that you can modulate it. And there are several examples in Down syndrome, in Rett syndrome, in Angelman syndrome, that you can, activate or replace the genes and have an effect, even in adolescence and adulthood. Now it does vary, like for example, and I hope I get this right, in Angelman, if you do it at birth in a mouse with Angelman, you kind of correct everything. If you do it later, you correct this, but not that. You correct some of the motor changes, but you don't correct some of the behavioral things. So it's different for every disorder. And that's why we have to do the studies. We don't know what it's going to be in PWS, whether it's going to be that we have a critical window or not. People were really surprised in Rett syndrome, because in Rett syndrome, girls are born fairly normal, then they regress. And so it was thought, okay, that's, once they regress, you can't ever bring them back. But at least in the mouse models of Rett syndrome, you can take an older mouse with Rett syndrome and kind of bring it back with a genetic therapy. So, so there's lots of examples now of the possibility, and that's why we need to go through. I'm not saying that it will, work in PWS, but we need to find out yay or nay.
Yeah. So there's lots of possibilities, there's lots of opportunity, but there's also a lot of work that we have to get done. So, thank you to everyone who's here today and who's been helping to support this research, because it truly is our community that is pushing this agenda forward, both with time and with funding. I want to just briefly share with you a really big opportunity that we have coming up, as well as the programs that and sessions that we have later today. So we have 3 sessions available today. Partnering for a Successful IEP. Katie Chambers is a principal, an administrator, a teacher, and a mom to a young boy with Prader-Willi syndrome. She's going to be talking about how you can really work with your school to make that successful IEP. And Dr. Dewis will be back again this afternoon. This session, the standards of care session, is really for our young kiddos, the ages 0 to 2. She's going to be really focusing in on care for that age. If you missed the standards of care for children and adults, it was recorded. It's actually already up on our YouTube channel, so you can go to our YouTube channel and see that recording there. And then this afternoon, if you're interested in learning more about the completed trial of DCCR, the Soleno team will be joining us and talking to us a bit about what's happening with their program and providing a clinical trial update. Last but not least, I really have to advocate for our advocacy session, which is taking place tomorrow morning at 11:30 a.m. Super excited about this. Our first potential treatment for PWS in more than 2 decades is currently under review by the FDA. As part of that review, the FDA is holding an advisory committee meeting, and we, the PWS community, have an opportunity to provide our input and ensure that our voice is heard. So in tomorrow's session, we will be discussing the upcoming meeting, what the committee will be reviewing, and how the community can participate and make an impact on the review and the decision. I highly encourage every member of our community to register for this webinar. Even if you can't attend, a recording will be made available. I will make sure that it is shared with you all tomorrow afternoon, so that if you can't attend live, you can still access this meeting and watch it, including in the recording and the presentation itself will be instructions on how you can provide your comments to the FDA. It's not very often that we, the community, get a chance to speak directly to the FDA about treatments. This is our chance. So come join us tomorrow to learn more about how you can do that. We have 2 clinical trials that are currently enrolling participants. So before we sign out today, I'm going to roll those videos, and I hope that I will see you later today or tomorrow.
If you or someone you love has Prader-Willi syndrome, you may be interested in a clinical trial open to people with PWS who are between the ages of 6 and 65. This trial will study the safety and effectiveness of an investigational medication and its impact on excessive daytime sleepiness, behavioral symptoms, and cognitive function. Clinics across the United States are currently enrolling participants, with more sites being added, and travel expenses to the clinic most convenient to you will be paid for by Harmony Biosciences. So, what's involved? Well, that depends on whether you are the person with PWS or the caregiver. If you plan to participate, there will be 5 scheduled visits over the course of about 4 months. And if you're a caregiver, you'll accompany the person with PWS to each of the visits. For caregivers, your support and participation will be important. For example, the first visit involves an initial screening and the start of a 2-week sleep diary. Making sure the diary is kept up to date will be the responsibility of the caregiver. The second visit includes nighttime and daytime sleep tests to confirm that the person with PWS is eligible for the trial. If eligible, the person with PWS will start taking their assigned study drug once daily in the morning after waking up. This could be the investigational medication or placebo. The dose will be kept in a diary, updated daily. Making sure the diary is kept up to date will be the responsibility of the caregiver. The next 2 visits are to assess how the person with PWS is progressing. At the last visit, the nighttime and daytime sleep tests will be given again, and then it's time to consider whether to continue into the long-term safety trial, where people with PWS will take the investigational medication and help researchers collect information. There will be no placebo during the long-term safety trial. Clinical trials are essential for the development of new treatments to potentially help people living with PWS. And every clinical trial starts with volunteers like you. To learn more about this trial, you can contact Harmony Biosciences at clinicaltrials@harmonybiosciences.com.